This call is now being recorded.
Hello, my name is Paulo Aecyo. I am a PhD student at State University of Campinas at Brazil, and in this talk I'll be presenting my PhD thesis, which has as title Genomic Basis of Local Adaptation of Neotropical bromeliad species along an altitudinal gradient. So local adaptation occurs when individuals of a population show traits that allows a higher fitness in its local environment than other environment. This process is guided by divergent selection, which selects different phenotypes in each population, but it may be hindered by gene flow, confounded by genetic drift, constrained by lack of genetic variation. Different genomic approach may be used for detect signs of selection through the genome. One of them is the RAD-seq, the restriction site of the associated DNA sequencing, which sequences regions close to a restriction site. However, because of the mechanic of actuation of this technique, it may be lost. It may lost some regions, maybe not analyzed some regions, and then lost some genes that which may be associated with adaptation. So for a more even analysis through the genome, it's possible to perform some whole genome resequencing in a medial coverage, which may be used for detect signs of selection through the genome. And it in fact has been used to study local adaptation to altitudinal clines, for example, in Salix brachysta and species of prunus, being possible to detect some signs of selections in genes related to DNA repair pathway, light stimulus, cold stimulus, and production of phenolic compound. And besides that, it's also possible to use the whole genome resequencing to analyze the pangenome of a species. So the pangenome concept arises first for microorganisms, that it was observed that when it was sequenced more than one individual per species, it was possible to observe that those individuals may share some sequences, which is called the code genome of those species. But also, some individuals shows exclusive sequences, which is exclusive to one individual or a group of individuals, and this sequence is called the accessory genome. Then so it's possible to observe that have only one genome is not enough to assess all the pool genic of a species. And also with the pangenome, with this resequence, it was possible to detect not only the present substance variation, this difference in sequences, but also observe some structural variations, and just as complement variations, translocations, but especially in fact the present substance variation. And this present substance variation, these PAVs, it is being observed in several species such as Oryza sativa, the rice, which was observed in 1,300 more new genes that was not previously described in the reference genome, but it was also possible to observe in Medicago trucatula, for example, 38,000 new genes that was not present in the reference genome. Then another question arises when we analyze these PAVs, which is the role, the adaptive role of these PAVs, which has been observed in crop species, the correlation of the PAVs and some resistance to several diseases, such as in sunflower, which was observed a correlation with the mildew disease. But those studies of pangenome is usually associated with, is being focused on crop species, and a little is known about the role in of the PAVs in speciation in wild species, for example. Well, we are going to use as a model the Pitcairnia flammea complex species complex, sorry. The Pitcairnia flammea is a bromeliad species from the Bromeliaceae family, the same family as pineapple, and it's a species complex which comprises around 6 varieties. This species is saxiclose. It lives over rocks. It's a allogamous species pollinated by hummingbirds with no clear adaptation for seed dispersion. This species is endemic of the Brazilian Atlantic forest, which is the second largest rainforest from the tropics. which is a highly heterogeneous environment and shows a high loss of habitat, as we can see here in this picture. Because of this, the high endemism of this region and this high loss of habitat, this region is considered a biodiversity hotspot for conservation. In a genomic point of view, Pitcairn flammea has a diploid genome with 50 submetacentric chromosome, showing a symmetrical karyotype, as you can see here, and with very small chromosomes and a very small genome size of 1.08 to 1.69 picograms. This species has been used as model for species studies, being investigated the hybridization among Pitcairn flammea and Pitcairn cocovadensis in some regions here in Brazil, being possible to observe the asymmetrical introgression among those species. And in a phylogeographic study, it was possible to observe that this species is highly structured, showing 26 haplotypes in 25 populations, and also 22 clusters in those 25 populations. It was almost as if each population evolved as a different lineage. And one of the reasons for that is the local adaptation. As you can see here, several traits is associated with local adaptation to altitudinal clients such as succulence, leaf area, and the distribution of abaxia of trichome in low and high altitude plants. So, however, little is known or nothing is known about the genomic basis of those local adaptations to those traits. Then we aim with this project to understand how the local adaptation to an altitudinal climate is leading to the genetic divergence of Pitcairn flammea populations. And more specifically, we aim to understand what is the role of the different evolutionary force in the genetic divergence and speciation of Pitcairn flammea populations, and what are the genetic bases of local adaptation of these populations, and how the SNPs and structural variations, especially PAVs, have contributed to local adaptation and divergence of those populations. And for that, we are assembling the chromosome level genome of those species, and we intend to, by resequencing some individuals, to assemble the pan genome and perform some population genomics analysis. So for that, we have collected 10 individuals and distributed in 8 populations through non-altitudinal climate, as you can see here, varying from sea level to 2,200 meters above sea level. And we've performed some sequencing for 2 individuals, one from Morro do Rangel, which is in sea level, as you can see in today's picture, and another is from Pico dos Marins, which is 2,000 meters above sea level. We've sequenced those individuals in a high coverage used by Hi-Fi, and we are assembling the genome of those 2 individuals. And besides that, we also have performed the omni-C sequencing for the Morro do Rangel individual, the run individual, being possible to detect that those sequences are distributed to the 25 pseudomolecules, as we can see here in this histogram. So we are still performing the assembly, but it's being possible to observe that those individuals has very small genome size, as you can see here, with 544 megabases or 535. There is some difference among those 2 genome, with an N50 of 2.5 and 3.5 for each haplophase of the run individual, the Morro do Rangel in sea level, and 4.2 and 4.5 for the Mar individual from 2,000 meters. And about the scaffolding, we've performed the primary scaffolding for each haplophase. So we've detected 1,800 scaffolds and 743 scaffolds to each haplophase, with a L50 of 20 and 18 scaffolds. However, we are still performing this analysis, so it's not finalized. But when we are performing the Brusco analysis to observe how many genes is being possible to detect, compared with the Embryophyta and Liliopsida, it's been possible to observe more than 90% of genes, 98.7 in Embryophyta and 93.4 for Liliopsida. And we also performed some previous analysis to observe how those genomes are being assembled, and observe if there is any difference among those genomes by now. So here we've compared the two haplophase of the run individual, and it was being possible to observe some structural variations. Some of them, such as these translocations in here, has a low percent identity, which may be associated with our unfinished assembly, but some of them has a high identity, such as here, which is being more clarified here in this insert. And we'll be able to observe some duplications and some inversion in these two haplophase, which may be associated with the high genetic diversity of these species. But it may be better investigated once we finish this assembly. We also have performed the analysis of comparing one haplophase of run and the pineapple genome, which is also a bromeliad species. And it's been possible to observe several structural variations among those two genomes, which may be associated with the time of divergence for those genomes, which is around 30 million years of divergence. Well, right now we are still performing the chromosome level genome assembly, but once it's finished, we are going to assemble the pangenome and the population and perform some population genomic analysis. So for assemble the pangenome, we are going to resequence 80 individuals in a medial coverage, and we are going to detect the structural variations by performing an iterative assembly. And those structural variations will be associated, will be positioned by analysis of linkage disequilibrium. We positioned the reference genome to assemble the pangenome. Besides that, we are going to perform the population genomics use these resequenced individuals, and we intend to perform some population demographic to study the population demographic history of those individuals, of those populations, sorry. And we are also going to study the genetic structure of those individuals by using both the core genome, by performing the FAST structure to observe how those populations are structured, but also to performing a PCA using the pangenome, especially the PAVs, to observe how those PAVs are structured among those populations, if there is any structure. And we are also going to study, to observe the force of genetic drift and the role of gene flow in this process. Besides that, we are also we intend also to perform selective switch analysis to observe signs of selection through the genome of these species, those individuals, those populations. And we are going we intend to perform some genome-wide association study to detect observe which genes may be associated with adapted phenotype previously described and previously analyzed, and then perform some associations to correlate these phenotypes, the genotypes, and the environments, which are also collecting data for that. And then we have as perspective, we intend to give insights about the onset of speciation process in one of the highest species regions of the world. So that's it. Thank you very much.